In the Gorleus building we have a lot of high-tech equipment for research and education. Let's visit a few of them. Almost all 8 institutes are located in this building, from astronomy to physics and from chemistry to mathematics. And in this hall we find no less than 3 very unique research facilities, from the measuring hall to NASEN to NMR, and we're going to visit them all. So, hey Bas, you're a researcher here in this measuring hall. Can you tell us something about the research done here?
So what we see here is actually many different research groups working in the same lab space. And we all have our own little islands. And we actually call them islands because they're almost literally islands. They are floating platforms and they actually float off the basement floor. And this is to isolate vibrations.
And what is unique of this measuring hall here?
I think there are 2 things really unique. First of all, the shared space and being able to collaborate easier in that way. And second of all, a bit more on the technical side, here we have a combination of very low temperatures and low vibrations. I think this is very unique. And on the other hand, in the case of Irene Groot, we have the combination of low vibrations, atomic scale and ambient and hot conditions.
Finding new ways to treat diseases is one of the core activities of Leiden University. With NASEN's powerful electron microscope, we try to understand what happens in cardiovascular diseases, infectious diseases and cancer. At the end of the day, we want to help improve treatments for these diseases. So, what happens here, Meindert?
Well, here at NASEN, we allow researchers from both the Netherlands and from Europe to come and use our machines to visualize complex biological structures that allows them to understand how those proteins work and how they execute their activities. And this is fundamental knowledge to understand how biology works, but also how we might become ill because of changes in those proteins caused by genetic mutations. It is also very important for us to improve novel drugs that may inhibit a particular protein, and with the information we can derive here with our microscopes, we can make those drugs even better, bind better and not bind to other proteins that we don't want to bind to.
And the microscope we have here downstairs is a quite special one. How does the quality of that microscope improve treatments?
So it is that minute information we can derive from our microscopes that helps the researcher develop new therapeutics or have a better insight into how biology works and how diseases might develop.
Oh, wonderful, very interesting. Thank you.